Hello everyone, welcome to AUA. I'm so happy that the Starmus International Festival continues in Armenia and at AUA in particular. We're celebrating science and art to infect young people with the spirit of discovery, and hopefully this spirit goes around the world. So we have had already 2 days with very interesting speakers. I would call them Starmus stars. And today we have the concluding scientific talk with Emmanuelle Charpentier, French microbiologist, geneticist, biochemist, Nobel Prize laureate in chemistry 2020 for the development of a high precision method for genome editing. While looking through the website of the Max Planck unit for the science of pathogens, I found this interesting quote from Aristotle. Excellence is never an accident. It is always the result of high intention, sincere effort, and intelligent execution. It represents the wise choice of many alternatives. Choice, not chance, and determines your destiny. So this quote provides us a clue to the success story of Emmanuelle Charpentier, the founding scientific and managing director of the Max Planck Unit for the Science of Pathogens and honorary professor at Humboldt University, Berlin, Germany. Emmanuelle founded this unit in 2018 to strengthen fundamental research on pathogens causing diseases in humans. She received her doctoral degree in France and pursued a scientific career in academic research institutions in France, the US, Austria, Sweden, and Germany. Very international career. Emmanuelle has devoted most of her scientific career to understanding fundamental mechanisms of diseases, with a particular interest in infections caused by gram-positive pathogen bacteria. She has been widely recognized for her groundbreaking research that laid the foundation for the revolutionary CRISPR-Cas9 genome engineering technology. She has received numerous prestigious international awards and honors and is an elected member of national and international scientific academies. She is also the co-founder of CRISPR Therapeutics and ERC Genomics with Roger Novak and Shaun Foy. So today, after the scientific talk with Emmanuelle Charpentier, who is going to share with you about her scientific discoveries and their application in different fields, and hopefully she will share some stories about her career growth to inspire you, the young generation, to choose the path of science and discovery. And after her talk, we are going to open up for questions. So I would like to ask you, the audience, to use the microphones on both sides so that we can hear not only we in the audience, but also the audience who is following our event online. Thank you, Emmanuelle. It's great pleasure to give the floor to you. Thank you for being with us.
Hello, everyone. This is a great pleasure for me to be here in Armenia, and thank you for the opportunity to give this lecture at the American University of Armenia. And it's really a great pleasure to be in your country, and I'm very happy to see young, motivated students, future talents of Armenia. So I understand that from the audience there are a number of non-biologists, and I always tend to go quite deep into the science. So I will try to make it short when it comes to the details of the science, but I may not succeed because I tend to also speak very long when the days are long, and since it's 11:40, I hope I will keep up also with the time. So actually, my talk of today is to a little bit show you the research journey that led to the CRISPR-Cas genome engineering technology, that has been extremely transformative for the past 9 years in the field of life sciences. But before, I would like to go to the acknowledgement. And as it was said in the introduction, I have been quite a mobile scientist. There are some reasons for this mobility. It's because sometimes you have to fight for your own destiny, in a way, which I thought for me was to remain a scientist when I started to be a scientist. And it's not always easy nowadays. You need to fight through, specifically when you're a macrobiologist, because my experience has been that in the field of life sciences over the past 20, 30 years, the fashionable topics were more with regard to cancer research for good reasons. Brain research for good reasons, and maybe now human genetics. A lot of biologists are very excited by the different applications of the CRISPR-Cas technology and other types of technologies that have developed very, very fast the past 10, 20 years. As a macrobiologist, you always refer to a little bit a kind of old type of research. And it was always actually initially a challenge for me to get grant money. But then I could always spin my grants by always ending up with always the same kind of sentences that will be that, looking, which was always the goal in my different labs, searching for new mechanisms that exist in bacteria, or going deeper in a mechanism that were already known in bacteria, would always be the unique, would be an essential strategy to be able to find new therapeutics, to develop novel therapeutic strategies. And also when one wants to work on bacteria, when one realizes always that bacteria are a large source of enzymes and RNA molecules that have also the potential to be harnessed as gene editing technologies. So this has always been, the reasons why at some point I was very successful in getting grant money. However, in the life of a scientist, I think the mobility is very important. Some scientists are very successful not being mobile. Others have to move around. For me, it has been very enriching, because I was always, confronted to new systems with new colleagues, where I learned to adapt fast, to also find my ways very fast, to apply for funds and have money very fast. And I think it's part also of the dynamics of the life science field. It's very competitive and it's international. And I think for me it has been very, very useful, even though it was not specifically, planned initially. So, as it was said, as a principal investigator, after returning from the US, where I did 3 different postdocs, I arrived in Vienna about 20 years ago. Then I moved to Umeå, and this is during this transition from Vienna to Umeå that the CRISPR research developed in my lab. So this was about, let's say, 14 years ago. And then from Umeå I moved to Germany in Braunschweig, and now I'm in Berlin. And the research was mostly done at Umeå University in Sweden, at the Laboratory for Molecular Infection Medicine, Sweden. Something important to say is that in science nowadays it's very difficult, specifically in the field of life sciences, to survive without, let's say, collaborations, or at least without having an environment that is multidisciplinary. So right now my unit is can be self-sufficient to a certain extent, because I have set it up with different expertise, with different scientists bringing different types of expertise, and also we are lucky to have a very good equipped lab. But collaborations are often necessary if one wants to go a little bit beyond and foresees what could be a potential of a research project. Therefore, for this research, actually, even though it really started in my lab, there were 3 collaborations. Collaboration with a group of Jörg Vogel with Cynthia Sharma in Germany. Then a collaboration with Martin Jinek, who was in the laboratory of Jennifer Doudna at UC Berkeley, and a collaboration with Eugenie Kounin and Kira Makarova at the NIH Bethesda in the US. And truly, I will not be here in front of you if young, talented students and postdocs will not have joined me for this challenging, or maybe not so challenging at the end of the day. We thought it was challenging, but it ended up to be maybe the easiest project to run in my lab for CRISPR. So actually, it started with some students who identified an RNA molecule that was very important for the understanding of the CRISPR mechanisms, and also one postdoc. And then after, really, the main work started with Elica Delcheva, who was a master's student coming from Bulgaria, and Krzysztof Szyliński, a PhD student coming from Poland. And I mention this because this is really an international team of young, motivated fellows, which can make a difference. So, having said this, the research on CRISPR started in my lab with our interest to work on Streptococcus pyogenes, that is a bacterium causing diseases in humans. It's actually responsible for a wide range of diseases, from mild infections to more severe infections. Here you can see the cocci of bacteria, colored in red for the purpose of the picture, invading pharyngeal epithelial cells, colored in green for the purpose of the picture. And so you have different sites of entries, and it's mostly the nasopharynx and the skin. Having said this, like every bacterium or every microorganism on the planet, specifically in the human host, the bacteria have to face a number of stresses that can be the immune response of the human host, and that can be as well ranging from a different range of acidity, heat, or other types of stresses. And you may imagine that in the human body, life is quite challenging for a bacterial cell. They really have to survive, and they have to adapt to their environment. And this is also important for Streptococcus pyogenes, that is a strict human pathogen. You don't find this bacterium anywhere else. It only colonizes humans, and 30% of us are colonized by this bacterium without developing diseases. And for sure, like also any microorganism, bacteria have evolved different types of mechanisms using different types of molecules. Specifically, for us, we focus mainly on proteins and RNA molecules that are involved in very complex regulatory pathways with really a lot of networking, and that ultimately regulatory pathways that are important for the spatial and temporal expression of factors that are important for the bacterium to cause diseases or to adapt to the environment and to survive. So spatial, because the bacterium can find itself in different sites in the body and will be encountering different types of stresses, and temporal, because during the course of infection, you have an invasive phase when really the bacterium enters more the body, and here for sure you have totally different types of molecular machinery entering into play that really allow the bacteria to cope differently with the different environments. Now, with regard to molecules of proteins and RNAs that can have regulatory functions in bacteria, here is just the genetic code that I want to remind you. Let's say maybe some of you have forgotten or but the DNA, right, is the carrier of the genetic information and contains the code of life and is transcribed into messenger RNA that are then translated into proteins. The proteins also rely on other types of RNA molecules, such as transfer RNAs or ribosomal RNAs, to build this protein synthesis machinery. And then what was discovered 30, 40 years ago is that RNAs can also have regulatory functions. These are in red, so-called small RNAs. They have different names in bacteria or in higher organisms such as humans or animals. They are called small RNAs, non-coding RNAs, small interfering RNAs, macroRNAs. And they have the ability to interact directly with the messenger RNA or the proteins and change the function of those small RNAs and proteins. There are different types of regulation. And when we started to work on CRISPR and on small RNAs in general in my lab, there was one interaction that was not described. So the idea is always to, when you do science, to hopefully maybe you work on something, hopefully you think it's going to be interesting. So in my case, I was always interested in infectious diseases because working on bacteria, because I was always interested in medical applications or biotechnology applications that could be developed through the bacterial work. And having said this, after you try to find a new mechanism. And in general, you're also interested in a mechanism that could be universal. So not specific to your specific bacterial species you work with, but maybe that can be also existing in other types of bacterial species or even other type of organisms. But there was one connection that was not identified up to the end of 2000, that was the idea of a small RNA that will interact directly with the DNA molecule to interfere with the function of the DNA and the fate of the DNA. And this is what CRISPR brings. And this is a technology. I mean, all our research on CRISPR led ultimately to this technology. It's called CRISPR-Cas9 technology, and it originates from a mechanism existing in bacteria. And it's composed of a protein that is called Cas9 that works like scissors. That's why you see the shape of the protein here as scissors. And that can be guided by an RNA molecule to a specific sequence on the target DNA. And it works like an RNA programmable scissors, so by which, in a way, one can modify any DNA of any cells and organisms in unprecedented manner, whereby if one would like to modify the DNA on a certain site, one can program this tool thanks to the RNA molecule, and that will work a little bit as a GPS system to find on the DNA the right portion to be cleaved by the Cas9 proteins. And the Cas9 protein cleaving the DNA molecule will trigger some repair systems in the cell, and the repair of this breaking of the DNA, depending on how the Cas9 tool is designed, will lead to either deleting a portion of the DNA or replacing a portion of the DNA by another portion of the DNA, or even changing a mutation by another mutation. So it can actually act on the DNA in different ways, and it allows to have a programmable system to anyway, in a way, rewrite the code of life. So it's a little bit like you use a software to correct a text, whereby you can change one sentence by another sentence, or one word by another word, or one specific letter by a specific letter. This is true. But this is the DNA. It's not on the text, it's on the DNA. Having said this, it's, if you look at all the genetics, the history of genetics, just to show you when CRISPR-Cas was important and at which step, In the 19th century, started the genetics with really the understanding of the origin of species, then the laws of inheritance, then the DNA that was isolated. And in the 20th century, the DNA that was shown to be the carrier of the genetic information, then the structure of the DNA that was determined, and then the genetic code that was deciphered. And for sure, then the need to, how do you say, to develop tools that would allow to trick the DNA to be able specifically, initially, to find out the functions of the genes, you know, the discovery that indeed we have the genetic code. There are a number of genes on the DNA of any type of organisms and cells, and the will to really understand, but what are the functions of those genes. And a lot of research done on bacteria and viruses, specifically focusing on molecules that were identified in bacteria and viruses, and specifically trying to understand how bacteria defend themselves against viruses, led to a large number of molecules that were discovered, and proteins, also RNAs, proteins that allow to clone DNA, all these recombinant DNA technologies, to sequence the DNA, to amplify the DNA. This is the PCR, which I think the public got aware of through the technology that is used to detect whether you are infected by SARS-CoV-2 or not. And then another types of different types of protein that would allow to really do specific gene targeting, what CRISPR does. With prior to CRISPR-Cas, two types of proteins that were called nucleases that were used in finger and talon nucleases. They can also cleave the DNA, but the way they were working was a little bit more cumbersome than CRISPR-Cas9. The programmability of these proteins, of these enzymes that cleave DNA, were a little bit more complicated than CRISPR-Cas. And when CRISPR-Cas entered into play 9 years ago, it really changed everything. And in my lab, the starting of working with CRISPR-Cas came from our interest in the class of small RNAs. Whether we have small RNAs in Streptococcus pyogenes that can regulate gene expression. So 20 years ago, we started with some small RNAs, and we found some small RNAs that had a role in Streptococcus pyogenes. Then we wanted to find out more small RNAs, and we found one small RNase that was actually encoded by a piece of DNA that was located next to a piece of DNA that was shown to code for a protein that was highlighted as a CRISPR protein from Streptococcus thermophilus. This is just to show you that people were already working on CRISPR, talented scientists who were already working on this system, and had already shown that this system was a quite complex system, yet sophisticated but very efficient at the end of the day, that would allow to, in a way, allows the bacteria and also other types of microorganisms named archaea to defend themselves against invasion by viruses. So obviously you know that you can be infected by viruses. You understood it by now. Every organism can be infected by viruses. Bacteria can be infected by their own viruses that are called bacteriophages, phages. And viruses can also be infected by viruses, which is the beauty of nature. And for sure, bacteria, like any organism, like us, we have an immune system that allows us to defend ourselves against the infection. And they have developed different types of systems. And the viruses of the bacteria, what they do is that they can inject, upon recognition of the bacterial cell, they can inject their own genome into the bacterial cell. And this will allow to produce the viruses that will ultimately kill the bacteria. They can also insert their own genome into the genome of the bacteria and bring new genes to the bacteria. So the bacteria have an interest in defending itself, and it has evolved actually different types of immune systems, like us. We have different types of immune system. We have the innate immune system and adaptive immune systems. So far, what was shown is that a large variety of innate immune systems, some of which actually were key to bring proteins that were used, like restriction enzymes that were used for molecular biology. And then came the CRISPR-Cas, that is the only adaptive immune system known in bacteria. So it's complex, and maybe I simplify it by saying that adaptive, it means that the system needs to adapt to the virus. The bacteria have to encounter first the virus, then there is something happening in terms of recognition of the virus, and it's in a way recognizing the virus by taking a bit of the piece of the DNA of the virus to insert this piece of DNA into the genome of the bacteria. And then after this, let's say, label of the virus can be expressed in the bacteria and used again when the virus wants to infect a second time to recognize the virus, and then there is a machinery that will recognize the virus thanks to this former recognition, and then cleave the DNA of the virus. And if you cleave the DNA of the virus cannot be produced, and it's a dead end for the virus. And that is the way the bacteria eliminates, can eliminate the virus. So we used a very, very thorough genetic and biochemistry approach, and very detailed, I have to say, to try to understand the mechanism, specifically the CRISPR-Cas9 mechanism, because you for sure heard the issue with the SARS-CoV-2, with the virus. It mutates a lot, and this is to be in the context of the arms race between the virus and for SARS-CoV-2, the human host. The virus mutates to try to escape our immune system, and for sure our immune system also tries to react, and so you have a battle, and the bottom line is that it's largely evolving. SARS-CoV-2 evolves a lot. So the phages of the bacteria, the viruses of the bacteria, is the same. They evolve a lot to try to escape, to not be killed by the bacteria. And the bacteria have evolved a lot of CRISPR-Cas systems to be able to fight a lot of viruses evolving. And so Cas9 is just one of these defense systems. And to make a long story short, we discovered at some point that indeed this RNA molecule that we had identified initially, that we called tracer RNA, which I talked to you about, was actually part of the CRISPR system for the specific CRISPR-Cas9 system. And that the way it was working is that the mechanism was such in a way that you could very actually easily program this system existing in bacteria into this CRISPR-Cas9 genome engineering technology that would allow to modify any piece of DNA of any types of organisms. This was important because at the time, and it's even now, the life sciences field is really witnessing a lot of developments. You may have heard as well, maybe some of you are not biologists, that you can sequence very easily your genome now. So it's thanks to the development since the beginning of the 2000s of very sophisticated, high-throughput sequencing technologies that allow to sequence genomes extremely fast and in an extremely deep manner. You have also a lot of technologies that allow imaging technologies that are very precise and that really allow to really visualize what is happening in a cell. You have a lot of other technologies that have been developed along the way. And when CRISPR-Cas9 arrived 9 years ago, it was really making sense for this technology because the people were just, the biologists were just expecting to have this technology to then, in coordination with all the technologies existing, to have finally a tool to understand the functions of genes and have multiple applications. So the success of the technology was shown very fast. after we published a science paper describing the last step of the CRISPR-Cas9 mechanism and offering the guideline how to use the technology, 6-7 months later, we could see, and it's very fast because you need in general to do the experiments, send out the article to be reviewed by your colleagues, and the article has to be published. But very fast, actually, it was shown that the technology was working in different types of organisms, in plants, in model organisms that are used in the lab to understand biology. So a lot of biologists work with flies that are Drosophila, or with fish, zebrafish, or with mice. And this you may know that there are lots of biology done in mice for research and development for also understanding the mechanisms existing in higher organisms and develop the therapeutics against also human diseases. And for sure, very useful as well in human cells, whereby the idea to really target specifically a gene, those technologies were not really well developed before CRISPR-Cas. And as I said, you can trick the DNA in different means. And what was interesting actually in this mechanism as well is that, I just say this because you know the evolution of SARS-CoV-2, and again I said this, the viruses evolve a lot, the defense systems, the immunity systems we have develop a lot. This is the same case for bacteria and the viruses of the bacteria, and this is really this evolution of the CRISPR-Cas9 itself that is very largely evolving. When you look at the DNA sequences of the CRISPR-Cas9 system, it's very much different from one bacterial species to another one, yet the mechanism of interaction of this CRISPR-Cas9 machinery remains the same. And it's really by looking at the diversity that we understand what was going on. And what I like about the CRISPR-Cas9 system is that you need to know that there are, I mean, obviously you have seen the past 50 years all the tremendous development in medicine and the possibility to have, you know, new drugs and new medicine. And it's a lot through the efforts of fundamental scientists, but after developers in industries or in biotech companies. And this all this research was mostly done, I mean, trying to understand human diseases was mostly done using models that were not really relevant to the clinical situations, because a lot of research has been done with mice. Just because up to maybe 15 years ago, it was not possible to really genetically modify the genome of human cells in the labs. So CRISPR-Cas changed a lot this possibility, and it also allows to study different types of cells. Because in biology, the problem as well is that people always work with specific types of cells. And for sure, we have a large number of different types of cells. And with also the technologies that have evolved the past 10, 20 years that allows to grow in the lab stem cells and other types of cells, this technology really makes very much sense, and it allows to really study the diversity of the organism. So it comes from evolution, very diverse system, and it allows to provide a tool that allows to really study the diversity of the biological world. CRISPR-Cas9 is not the only system CRISPR that has been, developed as a technology. Research on the CRISPR-Cas systems by my very talented colleagues led to the discovery of other types of CRISPR-Cas systems that are also used in the space, even though the CRISPR-Cas9 system from Streptococcus pyogenes seems to be the most used system. Now, when you think at large applications, and this is nearly the end of my talk, it's really changing. It has been transformative for the world of life sciences, because, as I said, it really boosts the fundamental research on understanding the functions of genes in different types of model organisms. In plants, it's very impactful because it allows to produce plant crops in a clean manner. The genetic tools that were used prior to CRISPR-Cas9 to modify plants, leading to these genetically modified organisms plants, were always technologies that would rely on strategy, which at the end of the day, at the end of the process of genetic modification, you would have the presence of foreign DNA that was used to, in a way, select for the plant that was genetically modified. But with CRISPR-Cas, you can produce a plant with a very specific modification, whereby if you sequence the genome of the plant, you could think that the modification could have occurred in a natural way. So there is no trace of foreign DNA. And this allows to produce clean plant crops. And it's an important strategy in the context of fighting the climate change, whereby there are a lot of researchers trying to anticipate what happens in the plants when they are stressed by stresses that maybe they may encounter in the future, and try to select plants that can resist those stresses. Then they look in the genome what is responsible of this resistance, and then they can engineer plants that in the future will be able to cope with those stresses and resist and still grow. So in this field, it's important. You have also all the field of research and development, because to develop new drugs or new types of medicine, you always come to the molecular level. So you always need to understand at the molecular level how a disease develops, and ultimately to find a molecular target for a new type of drug or a new type of medicine. And for this, you need to really do genome editing to try to understand the functions of genes, find new targets. And so that's why the technology is very useful to have all those screens that allow to find new targets for therapeutics. Or also, when you develop a drug, you need to test the drug in different types of models. So first in cell biology models, and then in animals, and then you have the clinical trials. And before to come to the clinical trials, you need also to have, let's say, means to really verify that the drug is working well. And CRISPR is very useful in this matter. Then very interesting, and to create these models of diseases that are mimicking the clinical situation. And then it's very useful as a gene medicine. So the idea to be able to treat genetic disorders, and this, it comes to gene therapy. And gene therapy using CRISPR-Cas can be a gene therapy that combines the correction of the mutation in a cell with cell therapy. So, for example, CRISPR-Cas was shown recently to work in patients having blood disorders, whereby the blood cells of the patient is, extracted from the patient, modified in the laboratory. Then you have all the safety and verification, and then re-injected back in the patient. And this has been shown to be working, and there are a number of proof of principles now with regard to the success of the technology as a direct medicine. What next? I mean, for sure, this technology also, what this technology has shown is that there is a large development of industry and biotech companies in nowadays that are doing a great work. With CRISPR, a lot of efforts from developers, clinicians in this space. The success of the technology relies on those scientists, developers, clinicians who show that the technology can be applied in medicine. And this, it's really something that I want to mention, because we are always, as basic scientists, being recognized, but at the end of the day, those working in the private space should also be recognized, in my opinion. But having said this, it always, nevertheless, that any discovery comes from fundamental science at the end of the day, or numerous technologies originate from really basic science. If you take the vaccine against SARS-CoV-2, I mean, for sure there was this conventional protein vaccines that have been developed against SARS-CoV-2, but you have the RNA vaccines from Pfizer and Moderna, for example, and those RNA vaccines, I mean, we can be very happy actually that it was developed so fast, because in general it takes longer. But the RNA vaccines, for example, it really, the strategy originates from really pure, I mean, combining all the expertise on vaccine strategy that has, all those strategies that have been developed over the years, but also the RNA component of them, I mean, the idea to vaccine with an RNA, a messenger RNA, and this comes from also very, very deep fundamental research on RNA molecules. So it's just to say that it's very important to maintain the fundamental science, and this is where we see, maybe for good reasons, right? I mean, a lot of young talents going more to industry and biotech, but I'm just here to say, because the past years we started to see a little bit a change, and specifically the past 2-3 years, I like to let's say challenge you as a young generation to really not forget that you have a huge responsibilities for the next generation coming after you. Whereas what I said, I mean, you study, you're an undergraduate student, and a couple of years later, maybe you're not a PhD student. And then if you choose a track in the academic field, you do a postdoc, but you know, time passes very fast, and 3, 4, 5 years later, you're already on the other side of the, you know, of the barrier, and you have to, lead a group. This was, you know, my experience. You're very young, and you have to lead a group. I started quite young with it. Maybe I was 31 years old, and then you have to organize everything. Your lab, you have to find the money, your loan, and it's here that you realize that, nevertheless, you know, then it's your turn to teach the young generation and to carry on with the science. And it's important because it's important that at least we maintain a minimal number of scientists and teachers at the universities, because otherwise, who's going to teach your your kids if you have kids? So it's very important to have this in mind, as much as the private sector may be very attractive, to really have this this responsibility to make sure that you maintain sustainability also for the future generations in really the educational system and pure fundamental research system. As much as I know it's difficult and it's not always easy to, but it's possible. Nothing is impossible. And I'm finishing my talk with it. Yeah. Thank you for your attention.
Can you hear me? Yeah. Okay. So my question is about the future of gene editing. So as I know, there are other types of nucleic acid-guided proteins, particularly Argonaute enzymes, which are single-stranded DNA-guided proteins. And if you try to compare, like, how they can compete with Cas system, if you try to like mention some pros and cons with those two systems.
Yes. So here, as far as I know, the basics of Argonaute proteins, actually, initially this was more based on, I mean, the Argonaute proteins initially, they are RNA-guided proteins that are targeting RNA. not the DNA, even though I think lately you have some specific Argonaute proteins that have been shown as well to target DNA. Indeed, actually, there were some also developments to try to see whether Argonaute proteins could be used as RNA-guided proteins to target the DNA, but so far CRISPR seems to be really the gene editing technology mostly used. But having said this, it allows me to say that future research on microbiology is quite exciting because obviously with SARS-CoV-2, I just say this because I think people I mean, you know how it is with infectious diseases. I mean, unfortunately, the fact is that if you have an outbreak happening in India or in Africa, our Western world does not react so fast. Here there was a fast reaction because this was a global pandemic. And the field of infectious diseases has suffered from the fact that industry has left the field of development of antibiotics. So for viruses, you don't treat viruses with antibiotics. You treat, you treat viruses with antiviral drugs, and otherwise you prevent viral infection with a vaccine. In bacteria, you have some also vaccines existing, but not against all bacterial species. But when you have a bacterial infections, you use antibiotics, and you have a lot of resistance against antibiotics. And there is really a need to understand better, and the field has a little bit, let's say, been impaired. The microbiologists who were my fathers and mothers in the field have now on their way to retire. And there are not a lot of microbiologists in the field. And I think it's important to have more microbiologists because in addition to understanding better infectious diseases and to have more tools to combat the future infectious diseases, bacteria or archaea, but specifically bacteria and viruses, are large tools of, large source of different types of mechanisms. So what you mentioned, Argonauts, actually it's more in the eukaryotic world, this system. But in any case, the thing is that, or in archaea, the thing is that it's one of the multiple systems that we will discover in the future, working more on macroorganisms to find all this, because it's clear that you have a lot of systems existing of RNA interactions like Argonaute or CRISPR that will be coming up and that will add to the genetic toolbox, whereby the future of gene editing, because this was your question, I believe CRISPR will be here for a long time, sure. But who knows? I mean, I will be glad that someone discovers, another mechanism that may be even better, because then, we can go faster with research and with the applications. But Argonaute, yeah, it's a very also interesting mechanism.
Okay, thank you.
Mm-hmm. Yes. Thank you for awesome presentation. What's your viewpoint about nanotechnology incorporation into CRISPR gene editing methods, such as, for example, non-viral nanocarriers, such as polymers, lipids, silicon nanoparticles, metallic nanoparticles, and a part of it, even using extracellular vesicles in gene editing?
So this is an interesting question because I did not mention it, but the technology is one thing, but actually maybe the most challenging is also the delivery system, how you bring the CRISPR-Cas system to the cells. And this system has to be efficient and safe. I mean, specifically if you think of using the technology for treating human diseases in an in vivo manner, whereby you will inject directly the technology, with the carrier system, the delivery system. So because you want to bring CRISPR-Cas to the larger number of cells existing, and you want that the delivery system is efficient and yet safe. So CRISPR-Cas has been developed a lot in different versions to be even more specific because depending on the piece of DNA you want to modify, you always have to change a little bit the recipe. But with regard to the delivery systems, you have different types of delivery systems. So you have the typical lentiviral systems. You can also deliver with, this is more for research purposes, maybe with a plasmid. So a plasmid is a kind of circular form of DNA that will code for the proteins and that will code for the RNA components of the CRISPR-Cas system. You can also inject directly the protein complex with the RNA component. So you inject directly the purified ribonucleoprotein complex. And this is done for example, to generate transgenic mice or knockout mice. And for sure, you have all the field of nanoparticles that this field has been very active since the technology that was, let's say, available to the biologists 9 years ago. So the field of nanoparticles with also the idea that you could have also lipid carriers. For the extracellular vesicles, it's also part of the delivery systems indeed that are studied. Interestingly, the extracellular vesicles are also actually, bacteria can also produce extracellular vesicles. There is also research in this field to try to understand better the vesicles that are produced in nature, and because it's a little bit part of one project that we had in the lab, to be able to again, this is again like what CRISPR-Cas does, you try to understand in nature how the systems are working, and if you understand the different types of vesicles, these are somehow tiny, tiny little cells that are produced by one cell, but have the properties, because they are very tiny, to bring certain molecules to the right site in the body. And by understanding better this kind of diversity of vesicles, one can also develop better delivery tools that are really, let's say, more specific to the site, in the body where the technology can be delivered. So you have a lot of development in this field. Mm-hmm.
Thanks.
Hi. Hi. Firstly, I am very happy to see you. My name is Mariam Shulaya. I am from Georgia, from Caucasus International University. I would be glad if I would see you in our university too.
Okay.
So, about my question, what's your advice for young researchers like me who are now establishing themselves in this field?
Yes, be patient. Go step by step. It's difficult because every path is unique among the researchers, right? And if I take my experience, for example, I knew one thing, I liked what I was doing, actually, but I knew that I could fail at different steps. And I was asking myself a lot of questions, like everyone else. But because actually what was driving me more, and I think this is where I say that one has to be honest with oneself. Academic research requires a lot of resilience and persistence, curiosity, and you have to face a lot of, battles in a way. Like in life, it's nothing more than life, but it's maybe an intense life, right? And it's a life where you cannot always control what is going on around you, because you cannot control whether, a project will work out. You cannot control in which environment you're going to find yourself, and maybe the environment around you is going to change and affect your own development, or maybe something will happen. So it not only depends on you, it also depends, it's a field where you're also judged. And you have to be ready to be judged. I mean, you're judged because you apply for grants. You have your reviewers, defining whether you're going to have this grant or not. Then for the papers, it's the same. So you have to be ready for this, but tell yourself that at the end of the day, you're all, sitting in the same boat, and that, in principle, you're not alone. But what is important is to, I think in life in general, is to, this was what I did in a way, I believe. I mean, at the time, I even don't know whether I was conscious enough to do so, but retrospectively, if I look at what I did, I was indeed challenging myself quite a bit. But challenging myself to, quite early on, to just see, okay, is it for me? You know, I mean, I like to be a PhD, I like to be a postdoc, even though each time I was like, can I really do so? No, maybe not. But then after I was like, well, actually, I'm doing it, you know. And that's why it's important to choose well your environment, because at the end of the day, you're not alone. You know, you're part of a community. And this, it's important to be surrounded by a community, you know, with which you can well interact, because it will help you as well. But the bottom line is that, you know, at some point, it's important to challenge yourself early on and then detect, okay, is it for me? Or maybe something else is for me. But if you like very much what you're doing, right, I think there are ways that you know, you can go ahead without looking so much left and right and give yourself a chance. In the academic world, nevertheless, this is what I think. More and more it will change. You know, at my time, we were in France, you had to study very fast, and there was an age limit to become a researcher. They changed this. I think it was good. And I think in the future as well, you know, and this is already what has happened. Some people can leave the academic field as, you know, maybe not too young and go to another space. So it's possible. But I would always recommend you to, if you like what you're doing and you're in an academic environment, to go in this direction. But then if it's not for you, it's not necessary to, you know, to lie to yourself and then you take another path. And again, for sure, I understand that in the academic world or in a university world, maybe the, you know, the salaries are not that attractive, but you need to think long term, actually, because you know, what is more important actually at the end of the day, and that I can tell you that for me as well, you realize that, you know, maybe when you're young, you want certain things right away fast, but then you realize that you have long years to live, and you want your living to be also enriching, intellectually speaking, and also that you can challenge yourself. And the freedom that you have to do your research, I think, is unprecedented. I mean, for me, I could do, as long as I could, you know, get funding, and for sure positions, but I could, you know, I could design my projects as I wanted. It gives a lot of freedom that you don't have anywhere else. So it was, you know, each time that I moved in different places, it was like I was bringing my house with me. People were like, wow, I'm like, yeah, my lab is a lab, the pipettes are the pipettes, my projects were the same. I had the same computer in front of me, just, you know, working with different people. But I was really bringing my own projects with me, and I think it's unique. You cannot do it if you work in the private sphere. So I think you have a lot of, you know, satisfactions, and intellectually, I think it's nice. It's my point of view.
Thank you very much.
Yeah. Hello, Emmanuelle. My name is Valentina Angarkhaeva. I'm from Irkutsk State University. It's in faraway Siberia. And I admire the life of Soviet biologist Timofey Versovsky, and from his book, Stories Told by Myself, he said about that science should not be approached with the ferocity of wild animals. And you know probably also about his life, and he liked his biology a lot, I'm sure. But he struggled a lot of difficulties with his fields, and maybe you have advice to the scientists that live now in the less freedom world, like in Europe, and what to choose: the truth about what's going on, or the safety.
Yes, I think I would choose the truth. The truth doesn't mean that it's unsafe, right? It's yeah, I mean, it's a very challenging topic indeed. Actually, what is interesting when you say truth or safety is that when I started, I just say this because I think it's the case for everyone. I mean, for me, maybe more actually. I was a typical French student of the 90s, you know, who liked to have, you know, maybe some types of security. Yet, when I studied, I was at some point at the Pasteur Institute, and I kind of understood that all, you know, the scientists who had gone further, they all had gone through a, you know, a postdoc in the US at the time, not specifically because one had to go to the US, it's because at the time it was common that the US had funding for postdocs, whereby in Europe the labs did not have a lot of funding for postdocs, and it was just only through fellowships. So it was maybe more, you know, natural to go to the US. And for me, it was really dealing a lot with my security. But obviously, I wanted to be a scientist. And when you want to be a scientist, you want to know the truth. I mean, the truth is relative, right? It's as much as you can make sure that in biology, that you have found something that is as close as possible to the true mechanism, because how do you say, the technologies can also limit you in finding exactly what really happens. Now the thing is that with regard to the struggles that one can face, either in the typical academic environment, or because you find yourself in countries where it's more, you know, difficult than others, then for sure it's unique paths, right? It depends on you, and it depends also on the environment that could evolve very fast around you and that you cannot predict. I will think that again, it's not, you know, it's not, you know, it's okay to choose safety. I just think that some of us in general have to maybe choose the unsafety, otherwise the world will not, you know, advance. This is my opinion. But this is a challenge in general of being a scientist. It's always this, you know, what do you choose? There is always a balance in a way.
Right. Thank you.
Hello, and thank you very much for the interesting presentation. My name is David, and the question I had is, there is a lot of interest and investment into the field, and there is no shortage of science fiction about the future of gene editing. You know, occasionally you read about some borderline ethical experiments in some countries, but you being in the field and the expert, how I would define the question is, assuming sometime in a foreseeable future, you know, 2 or 3 decades ahead, when we, by we I mean we as a civilization, have mastered the science and maybe the art of gene editing completely, what science, not fictions, but science predictions would you make for us? What new capacities humankind will gain? Obviously, the primary application is, you know, antiviral and life enhancement, but what other possibilities do you see already at this stage that are perhaps visible only to the experts and not to the general public? Thank you.
Yes, so I think I don't have the crystal ball, actually. The thing that is interesting is the ethical aspects you mentioned. There has been a couple of years ago babies in China, which actually have been, let's put it that way, are originating from a manipulation with CRISPR-Cas of the germline, and this has been heavily criticized and banned. Let's say I don't believe that so soon the technology will be used for human transformation, just because you have a lot of phenotypes that are not caused by only one gene, but by multiple genes. And then, you know, there is still the issue of safety that is not completely resolved even in human cells that are not human germlines. now, and also, you know, I believe, it's my point of view, I believe that the world will face a lot of challenges and maybe more important questions to solve than because, you know, than thinking that, you know, it's interesting to have a baby with a certain trait versus another trait, and also because you have other types of technologies that are used nowadays in any case, if it's to counter-select for a mutation that is causative of genetic disorders. In certain countries you have, you know, in vitro fertilization and the selection of clones against a specific mutation that are possible. Having said this, you know, the CRISPR technology is really at the beginning with regard, for example, to treating human genetic disorders. These are just proofs of concept, and it is expected that this will develop further with, for sure, all the regulatory aspects to have in place for safety measures when it comes to gene therapy, whether it's an ex vivo approach or in vivo approach, but also with regard to, how should I say, to the fact that, you know, the medicine, if you wish, if it's called a gene medicine, has to be accessible to a large number of patients. So nonetheless, there are still a lot of efforts to do for the safety and developing the technology further for more genetic disorders, and to see also over the years whether the clinical trials that have been successful with a treatment that occurred a year ago still remains, and for sure the democratization of you know, of the medicine, with lowering down the costs so that more patients can have access to this to these types of medicine. So you have actually also, CRISPR-Cas is a very good example of technology, and this is what we see in our world, right? A lot of technology is evolving extremely fast. And then having the system having difficulties to cope with the very fast development of the technologies, either for the applications that also require a lot of work to see how largely you can apply the technologies, and also all the regulatory aspects, and the fact that we are not always ready to follow, you know, the speed at which the technologies develop. But I'm pretty sure that in 10 years we will see more development in human medicine. The technology would have allowed us to understand better the mechanisms of life, and would have led to, you know, the identification of more mechanisms that can be harnessed for biotechnology purposes, biomedical purposes, or other purposes that are very useful for life. I mean, you have also CRISPR being useful for the production of biofuels, so also for, you know, energy purposes. So there are large applications, and I think we will see more and more these gene editing technologies, whether it's CRISPR or other technologies in this field. Mm-hmm.
Thank you.
Excuse me. Hello. It was very interesting. I'm not a specialist of this field, but it was fantastic to hear your presentation. My question is very, very brief. Do you use your technologies also for fetus, unborn babies, for changing the infection or whatever the unborn child get, to prevent their further suffering after the birth? Thank you.
Yes, so this, it's, yeah, in a way, as a preventive strategy. The technology, let's say, is really used to, right now, more to treat diseases rather than preventing diseases. But this goes along with what was actually asked earlier. It is possible that the future will see the development of the technology not only to cure existing diseases or existing genetic disorders, but that the technology may be used to prevent. But here there are some ethical questions, because, you know, do you act on the human germlines or do you act later on? and again, who has access to this technology, and is it safe enough, etc. The problem for infectious diseases, if you want to use the technology to modify the genome at some place in a certain gene that could help us coping better with infectious diseases, what will happen, and this is the case for the technology that has been used to, it's not very nice to say, but this is what actually is used as a language to design the CRISPR babies in China, was actually to mutate the T-cell receptor on the cells of the HIV virus. So with the idea that then you would have a resistance to HIV. The problem is that it was also shown that if you mutate this receptor, you may affect other types of, you may have other consequences. And so I believe that it's a little bit, I will believe science fiction, because the issue is that whatever, everything that is infectious diseases, what I said, it's you treat, you have a resistance, etc. So even if you have a mutation that you think may be important, it can be reverted. So I don't think it is really, possible to believe that, and again, who will have access to this, preventive CRISPR gene editing? So, maybe when we are not alive, you will have such applications, but I doubt.
To be mindful of Emmanuelle's time, I would ask for the last two questions, because we need to conclude the session. Thank you.
Thanks a lot. Amazing presentation. Since you already touched upon the risks and ethical considerations, I will move to the issue of popularization of the technique. Like before CRISPR, there was gene editing happening, but CRISPR made it more effective, more easy to use. And now you look on internet and you can buy your own do-it-yourself CRISPR gene editing kit, and then you have these genetic hackathons, hackers, and stuff like that. I wonder, what do you think is the potential of such popular events to actually come up with groundbreaking technologies? Are these just like games that people enjoy and have fun with, or you think popularizing it to this level may actually lead to some serious technologies coming up?
Yes, so it's another very, very interesting question. Let's put it that way. This reminds me a little bit that when I was a kid, there was this kind of, how do you say, little games for kids, how to be a little chemist, when you have something red and yellow and you mix, and etc., etc. In biology, you really need to have a very, very, very well-equipped garage, basement, to really do high-level research that would allow any individual to really go further with the technology. This is my point of view. You really need to have a lab setting. That is my point of view. So at the end of the day, it would have to be someone also a very qualified scientist. Maybe some very rich qualified scientists can open a lab in the, privately in a way, but then it's an official lab and it's not really a private endeavor. And there was this, these stories of people injecting themselves, but I think it does not go very far, and this is my point of view. Mm-hmm.
Last question.
Hi, thank you for a good presentation. And I have one question. What types of application can CRISPR be used for in today's world?
In today's world, the application of CRISPR?
Yes.
So actually, I mean, if I go beyond what I said, so for example, for the engineering of plant crops, but it's interesting what you say right now. You know, if you ask me right now, the issue, for example, in Europe is that there are some regulations with the European Court of Justice that has decided that plants that will be modified with CRISPR are still labeled genetically modified organisms. So they have to go through all the regulations. So basically, it's not allowed to commercialize CRISPR modified plants, whereby whereas in the US or in the Asian countries, it's possible. So which means that indeed you start to see CRISPR genetically engineered plants on the market in the world. And that, for sure, at some point, I think Europe may change because there was a lot of pressure on the scientists and they realized that they may lose economically speaking. But for example, it's an example that came actually quite fast, a real example of applications that are real. And as I said, patients with sickle cell disease and beta thalassemia blood disorders, that are a few patients who went through clinical trials and are living, safely without the need of blood transfusion since they have, received this gene therapy methodology using CRISPR. So these are effectively things happening now. Mm-hmm.
Can I really quickly, I'm so sorry, we promised to get you out of here. So earlier this year, we had the honor of hosting Dr. Artem Pataputian, the first Armenian Nobel laureate, and he shared with us his, the experience when he got the phone call that he was getting the Nobel Prize. So can you really quickly just kind of share how did that feel? What was that like?
Yes, so actually, so this story is quite specific because very fast it was clear that there would be a large impact. Even for me, I knew this in a way, but for sure, you as maybe, I mean, you know that something is going to be big, but you don't allow yourself to say. But very fast, the people were telling, I mean, when we published a science paper, people were like, yeah, I mean, already the Nature paper that we had published before was, clearly would lead to something even better. And the people were already starting to say, you have a great story. But then, so I was prepared in a way. What I want to say is that it's been, when I received it, it was already, 8 years, even more, that I was prepared at some point. And because, I received a lot of prizes, and also with my ex-collaborator, Jennifer Donna, it was clear that at some point it will come. So in my research institutions, it was a bit something I didn't like so much, but in 2015, they were ready, and and then they were less ready because they were like, wow. But actually, the specific year, I was more the gut feeling, so I was more ready. So I was in my office, and in a way, I mean, I just say it because it's clear. I don't want to lie. I was like, I have this feeling that maybe. And then, I received, I was living in Sweden, right? So I know how a Swedish, a phone number looks like, and I know that it was a call coming from Sweden, and then it was clear, and but I could not tell anyone before it's announced, publicly, and I left my office, and then I came back, and then the people saw me quite relaxed, so they said, it's again, it's not this year. And then they were very surprised when they heard it like 40 minutes later. But for sure after, it's really a storm because it was also happening during the pandemic time, and so I was not used to give, interviews as I used to give before, and I was less used to interact like this spontaneously. So I had to be to be ready for the challenge, because after it's a challenge of a couple of days. Yeah. Mm-hmm. Yeah.
Thank you. And to use the opportunity that Narek asked the last question. I want to thank Narek because he was instrumental in organizing the series of wonderful talks and helping all of us to experience this great hours with the Starmus stars. Emmanuelle, thank you so much for this opportunity. And as always, I think greatly talented people are so approachable and modest. And thank you for that. And I hope that our students would feel comfortable approaching you or asking more questions, maybe, just informal chats after we're done with this session. But thank you very much. And I just want to emphasize that I'm so happy that our university have chosen the strategic direction of bringing different disciplines together.
It's very important.
Data sciences, bioinformatics, health sciences, data-driven life sciences. So this is, and your talk and your experience demonstrates the importance of this bringing everything together for, leapfrog development and addressing global challenges. Thank you very much. And
thank you.
I want to encourage our audience to continue enjoying these moments that Starmus Festival is bringing. Please know that today we're starting to have the booths, the science booths in the opera. There will be some concerts, and I'm happy to say that our provost and his band will have a concert today. So everyone is welcome to join us in this celebration. Thank you very much. Thank you, Emmanuelle.
And one final word for me, because